So our study addresses a long-standing question in tumor virology, namely what is the structural basis for the multiple and dominant interactions of small adenoviral oncoproteins? In other words, how do small viral proteins win? So to give you a bit of a context to that, cancer was once a molecular black box. And the key that opened that was actually revealing the interactions of small DNA tumor virus proteins. 20 proteins to take over a human cell. Remarkable. What makes it even more remarkable is most of them are less than 20 kilodaltons, and they have no sequence homology to human proteins. And after 40 years, none of their complete structures have ever been solved. So what if we could reveal their structures and their principles of design? Would we be able to come up with new molecular structures that disrupt the key protein interactions? And that's what we set out to do in this study. The one we chose actually to do it with is a protein called E4ORF3. Now this is a really challenging one because it actually forms a polymer, an incredible polymer structure in the nucleus. But it binds and disrupts 4 different tumor suppressor complexes, which are about 5-10 times its size. So the question is then, what is the structure of ORF3 and how does it simultaneously assemble into a nuclear matrix that inactivates multiple tumor suppressors? Horn, a postdoctoral scholar in my lab, figured out how.
To solve the structure, I have to break down the polymer into smaller subunits. I have identified oligomer mutants by making mutation for asparagine to alanine at 82 position of the proteins. This enabled me to isolate soluble proteins, obtain crystals, and solve the structure at 2 angstrom resolutions. So this is the first complete adenoviral oncoprotein to be solved. It doesn't resemble any cellular protein that can assemble polymers or any protein that involved in the tumor suppressor pathway. It is a dimer. It has a central beta core formed by the beta sheaths and is sealed at the front and back by the C-terminal tails. How does E403 dimer units assemble a polymer using this structure? What we found is the oligomer mutants cause the C-terminal tail to be in a closed state by binding to the beta core and prevent higher order assemblies. In wild type 403, the C-terminal tail can swing out and expose the beta core, in which 2 E403 dimer units can exchange the C-terminal tail reciprocally to generate a linear structure. Or they can also swap non-reciprocally to generate a branch structure. So in summary, E403 assemble a nuclear polymer by using a repetitive unit in a non-repetitive way.
So the model predicted that ORF3 would assemble a very novel macromolecular structure, a novel polymer assembly. Well, to be able to see that kind of structure, you need to use electron microscopy. Well, adenovirus has been studied for the last 40 years, but this structure has never been seen in the nucleus. The polymer is invisible. So to reveal it, we needed to use new techniques and technologies in electron microscopy. So what Horn did is he fused mini-SOG to ORF3, and we genetically engineered a virus that expresses this. When you shine light on it, it now produces singlet oxygen that creates an electron-dense material across the polymer, now revealing the nuclear structure of ORF3. And you can see as you look inside the fiber, the cable, that there's no repetitive structure. It's a series of threads that can twist and create new and remarkable 3-dimensional shapes. So in order to visualize the 3-dimensional matrix, we used a very special kind of electron microscope. And you slice it, slice by slice, 150 times, and you scan each one. And then you reconstruct the entire volume. And it gives us a holistic view inside a human cell that's infected with a virus that's never been seen before. And what you see is that the ORF3 polymer is bending and weaving and twisting through the nucleus. It separates cellular chromatin from viral replication domains. It branches and it captures cellular tumor suppressors, all in 3D. So the question then is the assembly of ORF3 into this remarkable polymer required for its biological functions? And the answer is a resounding yes. ORF3 dimers that cannot assemble into the scaffold are completely defective. But why? And what we reasoned is that individual E403 dimer units could actually have a very low affinity for its cellular targets. However, when ORF3 assembles into a polymer, now individual weak interactions are offset by having multiple binding sites. Okay, well, nice theory, but how do you test something like that? Well, lamin is actually a cellular protein that forms intermediate filaments. So what we did is we fused an ORF3 dimer to lamin and used it to assemble a novel protein superstructure to see if it creates new avidity interactions with PMO. And the answer is yes. And in fact, this new structure, it's a synthetic protein really, creates these cylinders that go through the nucleus, and they bind and disrupt the PML tumor suppressor. To date, our understanding of multifunctional protein interaction hubs is derived from cellular proteins. And there's two main mechanisms. So what ORF3 does is flout both of these conventions, and it uses a novel paradigm. It uses a small ordered protein structure that assembles in many different ways to create a disordered three-dimensional structure now that can capture many different tumor suppressors. So in summary, ORF3 forms a dimer. We revealed its structure. And upon dimerization, it creates 4 new binding sites that allow it to self-assemble in many different combinations to form a non-repeating polymer and an emergent multifunctional hub. This creates an avidity matrix in the nucleus that captures many different tumor suppressors and silences p53 target genes. It also separates viral replication domains in 3 dimensions from cellular chromatin.